To gain a better understanding of how viruses actually infect cells, and specifically how viruses infect human cells, let's take a look at the way that our HIV actually gets inside the human cell, the cells found in the immune system of the human body, and how the HIV essentially replicates and creates more HIV systems. So HIV is a human virus, is a virus that infects human cells, and it stands for human immunodeficiency virus, and it can basically lead to AIDS. Now, basically, the way that the HIV system looks like is as following. We have our single-stranded RNA molecules shown in red, found inside the protein capsid shown in green. And inside that capsid, we also have different types of proteins. We have a specific type of protein known as reverse transcriptase, which is basically a protein enzyme that acts to transcribe RNA into DNA, as we'll see in just a moment. Now, the HIV also contains the purple lipid-rich layer, and this purple lipid-rich layer contains protrusions, which are basically receptors which will bind to the protein receptors found on the cell membrane of the human cell. So in step number 1, we basically have the HIV, the virus, that approaches the cell membrane of our human cell. And in step 2, we have the binding process taking place. These purple protrusions basically bind to the receptors, the protein receptors found on the cell membrane, and specifically they bind to the protein receptors called CD4 and CCR4. Now, once that binding takes place and the HIV recognizes this specific type of human cell, we have the fusion taking place, the fusion between this lipid-rich layer found on the virus and the cell membrane of that human host cell. So in 3, once bound, the lipid-rich envelope of the virus fuses with the plasma membrane of the host human cell, injecting the contents, including the proteins as well as the single RNA molecules, into the cytoplasm of that cell. And next, we move on to step number 4. In step number 4, right within the cytoplasm of that human cell, these reverse transcriptase enzymes basically transcribe the RNA into DNA, and we form the viral double-stranded DNA as shown in blue. So we form 1 and 2 strands, and then they basically fuse to form our double-stranded DNA, the viral double-stranded DNA. Now, in step 5, we have the viral double-stranded DNA then basically travels into the nucleus of that cell. And in the nucleus of that cell, another special type of enzyme known as retroviral integrase, or simply integrase, acts to basically integrate or incorporate that viral double-stranded DNA shown in blue into the DNA genome of that human cell. Now, once that actually takes place, we move on to step 6. So in step 6, the cell then transcribes the viral DNA back into the viral RNA, as well as viral mRNA that can be used to synthesize viral proteins. So in step 6, we see that the ribosomes of our human cell basically use the mRNA, the viral mRNA, to produce special types of proteins that need to be used by the new HIV system, by the new virus, the HIV system. Now, we move on to step 7. Once we synthesize all the proteins as well as the protein capsid within the cytoplasm of the cell, all these important contents then basically move towards the cell membrane of the human cell, and they begin to push on that cell membrane to form this lipid layer as shown in purple. So the single strand viral RNA as well as the viral proteins move towards the membrane of the cell and begin to push outward on that cell membrane. And finally, in step 7, once our system is outside, the virus matures into our HIV system, the human immunodeficiency virus that can go on to infect other cells within the human body. So this is basically the life cycle of HIV. It's the method by which the HIV system infects the human cells and uses the machinery, the organelles of the human cell to basically replicate our HIV system to produce more HIV systems that can go on to infect even more HIV, even more cells. And this can basically lead to a syndrome known as AIDS.